Welcome. Today, we're exploring the complex world of glioblastoma, a highly aggressive and lethal brain cancer. For a long time, research has zeroed in on genetic mutations. However, scientists are now looking deeper into the complex interplay of genomic and epigenomic factors, particularly the three-dimensional, or 3D, architecture of DNA. This expanded view is uncovering new ways to understand and potentially treat this devastating disease. Our DNA, if stretched out, would be about 6 feet long. To fit inside a tiny cell nucleus, it undergoes intricate folding. This 3D organization isn't random. It brings distant genetic regions close together, creating functional hubs that coordinate normal cell processes. Groundbreaking research, like a recent study from Weill Cornell Medicine, has revealed that in glioblastoma, these 3D hubs become hyperconnected. Cancer-causing genes cluster and coordinate with other genes, some previously unknown in glioblastoma, to fuel tumor growth. This 3D reorganization can sometimes be a more significant driver of brain cancer than individual gene mutations. By targeting these hubs, instead of just single mutated genes, we might find new ways to disrupt the cancer. Epigenetics, which are modifications to DNA that don't change the sequence but affect gene activity, are also crucial. They shape the 3D genome architecture. In glioblastoma, these epigenetic mechanisms are often disrupted. For instance, mutations in genes regulating epigenetic modifications can alter chromatin structure and, consequently, the genome's 3D organization, leading to abnormal gene expression that promotes cancer. Pediatric high-grade gliomas often show mutations in histone proteins that lead to such global changes. Then there's the non-coding DNA. A large portion of disease-linked genetic variants are found here, especially in enhancers which regulate genes. Genetic variants or structural changes in these regions can dramatically alter the 3D genome landscape. This can lead to enhancer hijacking, where structural variations bring enhancers too close to oncogenes, wrongly activating them. We also see disruption of insulator elements that normally create boundaries between chromatin domains. Losing these boundaries can cause improper gene activation. Long non-coding RNAs also play a role, influencing chromatin structure and gene expression, and their dysregulation is increasingly noted in glioblastoma. Adding to the challenge, glioblastomas are extremely heterogeneous. Cells within a single tumor can have different genetic and epigenetic profiles, driving resistance to therapy. Advanced techniques are now being used to map this heterogeneity across a tumor in 3D, linking genetic subclones to patterns of chromatin organization and gene regulation. Understanding this spatial variation is vital for developing treatments that can target all cancer cell populations. So, what are the therapeutic implications? This growing understanding is paving the way for novel strategies. We're looking at targeting these regulatory hubs to disrupt oncogenic programs. Epigenetic reprogramming aims to correct aberrant modifications or restore normal 3D chromatin architecture. Researchers are also identifying structural vulnerabilities, like enhancer hijacking, that can be targeted. There's even research into small molecule inhibitors that can interfere with non-coding RNAs to inhibit tumor growth. The field of 3D genomics in cancer is evolving rapidly, providing unprecedented insights. This multifaceted approach, moving beyond a singular focus on mutations, holds significant promise for unraveling the complexities of glioblastoma and ultimately developing more effective treatments. The discovery that these hyperconnected 3D hubs might be a common feature in many cancer types further highlights the importance of this research. Thank you for listening.